Hello, everyone, and welcome to today's webinar from Blind Spot to Biomarker, Transforming Brain Health Monitoring with ImmunoPCR. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's web seminar is presented by presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, please visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you happen to have any trouble seeing or hearing the presentation. Now, I would like to welcome our speakers for today's webinar. Dr. William E. Haskins, CEO and co-founder of Gryphon Bio and Owl Therapeutics, and Aleksandra Hemm, product manager, Thermo Fisher Scientific. Aleksandra, you may now begin your presentation.
My name is Aleksandra Hemm, and I'm thrilled to kick off this webinar today. The title of this webinar is From Blind Spot to Biomarker, Transforming Brain Health Monitoring with ImmunoPCR. I will present our Thermo Fisher platforms, and Dr. William Haskins from Gryphon Bio will talk about how ImmunoPCR technology is important for new biomarkers and clinical assay development. Today's agenda is introducing the biomarker discovery and validation platforms that we offer, ImmunoPCR platforms and how they work. Our focus will be Neurofilament Light as a biomarker and why is it important. And then we will focus on the Neurofilament Light immunoassays. Thermo Fisher now has the most comprehensive tool set to support biomarker discovery, validation, and translation. The Olink assays enable profiling thousands of targets and help to identify potential biomarkers of interest. With the follow-on studies, with a large number of samples and fewer biomarkers, are typically move more into routine analysis, where platforms such as Luminex and also ELISA and ProQuantum as single targets technology are used for protein quantifications. In today's presentation, I will be focusing on the ProQuantum as an ImmunoPCR platform that uses qPCR to quantify proteins. This platform shares many benefits with Olink platforms in terms of sample input sensitivity, but also is available to perform up to 40 samples in duplicates. The assay workflow is homogeneous with no wash steps and 2 hours from sample to result. Olink proximity extension assays technology enables high sensitive and specific protein detection. It works by using antibody binding and DNA amplification to quantify the proteins of interest. The assays are scalable. That means that you can quantify from 5 to more than 5,000 proteins within a single platform. And the workflow are efficient because it has a streamlined workflows and also very well understood technology. On this slide, we will see the ProQuantum high sensitivity assays that are based on proximity ligation assay technology. On the first step, so how this assay works, we have first step is the analyte binding. Similar to the sandwich ELISA, the assays utilize a pair of antibody, each of those pair being to a different epitope of the epitope site of the analyte. Each of those antibodies is pre-conjugated to DNA oligonucleotides. One antibody is conjugated to a 3' oligo. The other antibody is conjugated to a 5' oligo. When the antibodies bind to an analyte, it brings the DNA ends into close proximity. Step 2 here is with the use of complementary splint oligo and DNA ligase, the two strands ligate together to generate the DNA template for amplification. Following, we have a melting on 95 degrees and inactivating the ligase, the analyte and antibodies. And now we have the simple DNA assay with 40 cycles of qPCR. The amount of DNA measured after each cycle is measured by a fluorescent dyes, and with increasing of fluorescent signal is in direct proportion to the number of qPCR product generated. What are the advantages again of this technology? This technology uses the specificity of the matched antibody pairs, but also the sensitivity and the broad dynamic range of the qPCR. A low sample consumption is also, so uses 2 to 5 microliters, has a fast and easy workflow, so no wash steps, 2 hours from sample to answer, broad dynamic range, and very important, it's an open system, so no instrument is needed to run the assay, and also include a cloud-based software for data analysis. So in this slide, you will see our per quantum high sensitivity offer. We offer human and mouse targets. Highlighted in bold are the neurobiology research targets that are very important, especially our latest edition, the NFL, that plays a really remarkable role in the neurobiology research. We also offer custom development services that can be found on our webpage. On the next slide, you will see why the neurofilament light is so important. What are the key functions? It facilitates the structural support of the axons and also maintain the shapes and the integrity of the neurons. If elevated levels are expressed of NFL, then it can be expressed in cerebrospinal fluid or blood that indicates that there is neuroaxonal damage. On the next slide, you will see that this biomarker is important in neurodegeneration, which happens in Alzheimer, Parkinson's disease, multiple sclerosis, and it's important for early detection of disease progression and treatment response. Also important is the NFL in traumatic brain injury to monitor the injury severity. In Vitrogen NFL immunoassays, we offer a very flexible platform, including the coded ELISA, which can be run on microplate readers, for quantum assays used on a qPCR systems, and also Luminex multiplexing assays that use the Luminex XMAC technology. We offer Neurofilament Light, as mentioned before, for quantum assays. You will see that below, under specification, we offer the same specification, high sensitivity, assay range, sample volumina 2 to 5 microliters as offered for the rest of the per-quantum assays. On the right-hand side, you will see the comparison between the per-quantum and Simoa, which is the best high-sensitivity technology most used on the market. You can see that our assay per-quantum is highly consistent and aligned to the Simoa technology, with R squared being equal to 0.98. Just for a reference, R squared 1 means 100% identical. So we are really well aligned to this technology. On the next slide, you will see what are the benefits of per-quantum versus traditional ELISA. As I mentioned, a couple of them are the sensitivity is important. So we detect proteins in pentagram range. Sample consumption is 2 to 5 microliters. Time to result is 2 hours. And the dynamic range is much broader, and there are no washing steps for ProQuantum. NFL assays, all 3 platforms that we are offering, so single target technologies, ProQuantum and ELISA, and as well as multiplexing technology, ProCardioplex, have been able to detect NFL in CSF, plasma, and serum samples, as you can see in the figures below. Also, all our 3 platforms are highly consistent, with R-square being over 0.9. That means that user can easily switch from a high plex usually to a lower plex, such as with single plex, such as ProQuantum or ELISA assay. And here I'm going to talk about the ProCardioplex assays that use the Luminex XMAC technology. We also offer multiplex solution for neurobiology research. This is offered with our ProCardioplex Luminex assay. As you know, the neurology diseases are very complex, so sometimes more targets are requested to be measured at the same time. This is possible with ProCardioplex. So we offer not only the NFL in this platform, but also other important neurological targets. This assay is available in simplex assays, pre-configured panel, but also very flexible mix and match panels where a user can choose their analytes of interest. It has been validated for all the different sample types needed, and also what we offer a free of charge that data analysis app, but also a custom panel designer such as panel configurator for users to choose their targets of interest. ProCardioplex technology employs the Luminex XMAP for highly specific multiplex biomarker detection using color-coded bits and fluorescent bit signal measurement, where target analytes are captured and detected through a sandwich complex similar to ELISA with capture and detection antibody. There are 2 lasers, one for quantification and one for specificity. Below, you will see all the Luminex instrument systems that are available on the market with the newest edition of the IntelliFlex and also XMAP IntelliFlex DRSE. And here is an example of our customer from Washington University, Borsky Center, that used the ProCardioplex for precisely quantify levels of 18 neuronal biomarkers in pediatric cerebrospinal fluid samples, as shown on the left side, and as well as known metastatic versus metastatic plasma samples on the right-hand side. So we not only offer target in neurology research areas, but also cover with our portfolio targets from more different research areas such as immunology, cell signaling, cardiovascular diseases, and endocrinology, and many more. And with this slide, I want to thank you for your attention and want to hand over to Dr. William Haskins from Gryphon Bio.
All right. Thank you so much, Aleksandra, for that very informative presentation. Before we move into Dr. Haskins' presentation here, we do have a couple of poll questions that we would like to call your attention to. The first that we have here says, was the presentation helpful for your research? If everybody could take a moment to select their response and submit, that would be wonderful. I'll give you about 15 to 20 more seconds here. Okay, and let's go ahead and move into this second poll question. This one says, would you like to receive more information about the Thermo Fisher presented immunoassays and be contacted by a sales representative? Again, I will give you about 15 to 20 seconds to answer this question here. Your feedback is very important to us. Okay, thank you all very much for your participation. Dr. Haskins, over to you for your presentation.
Okay, good morning. My name is Will Haskins. I'm the CEO and co-founder of Gryphon Bio, and I'm going to talk to you about how we're working on ImmunoPCR platforms from Thermo to transform brain health monitoring. And I want to thank the opportunity to present today, as well as thank you for your time. Here's a disclaimer. So you just heard a nice introduction to some of the technologies from Thermo from Aleksandra. I'll provide, again, a very brief introduction to ImmunoPCR in general, as well as traumatic brain injury and Alzheimer's disease, which is the focus of Gryphon Bio. And then I'll follow that with a discussion about how we're using ImmunoPCR in two different ways for brain biomarker discovery and clinical assay development. In particular, the proximity extension assays, or PEA-based proteomics assays for biomarker discovery, and the proximity ligation, or PLA assays for clinical assay development. And then I'll wrap up with some thoughts on future directions and potential impacts on how we can transform brain health monitoring. So I know some of this is redundant from the introduction from Aleksandra, but suffice to say, ImmunoPCR does combine the specificity of immunoassays with the sensitivity of the polymerase chain reaction. And I have a mass spectrometry background, so it took me some time to adapt to this, but it's got some definite advantages for the types of projects I'll be showing you today. Essentially, the cartoon on the left shows how antibodies can be used to provide high specificity detection of a protein biomarker. And then the cartoon on the right shows that if you label antibodies with DNA, you can amplify up the DNA on the antibodies bound to the biomarker to get a signal. And so this is a really clever way to make detection of your biomarker targets. So we're really focused on traumatic brain injury and Alzheimer's disease because these really drive the global brain health crisis. There's actually more than 100 million new patients each year globally for TBI and AD. And unfortunately, that means that you or someone that you love is likely to suffer from one of these diseases or some adjacent brain condition. It's actually about 1 in 5 of us will get traumatic brain injury by age 65, and 1 in 13 of us will get Alzheimer's disease by age 65, with higher numbers if you have risk factors. So, for example, traumatic brain injury is a risk factor for Alzheimer's disease, along with, of course, age and various genetic factors. So there's a real need to come up with better prevention strategies. And to do that, we really need to monitor and manage the patients, especially since the world is getting older. One way we think about this at Gryphon is that you have cognitive impairment in TBI and AD patients that can develop from impaired clearance of brain cell debris by microglia and other clearance mechanisms like the glymphatic system or lymphatic system. And this cartoon on the right was simply showing that you can have a microglia will eat or phagocytose the debris or pathogens in the brain And that's a beneficial thing. That's beneficial clearance. But your brain health is really a balance between that beneficial clearance and kind of the harmful accumulation of debris like p-tau and amyloid beta. So we want to monitor your brain health and this balance between clearance and accumulation. Up till now, the main symptoms of cognitive impairment have been used for diagnosis and treatment. So that would be headaches, confusion, and memory loss, for example. But these clinical measures are generally subjective and qualitative, so there's a lot of room for improvement. So by quantitatively measuring biomarkers in different biofluids like blood, CSF, and capillary blood, we can use this as a surrogate measure of cognitive impairment, and we can do this quantitatively. Moreover, understanding how that debris is cleared from the brain from different cell types like astrocytes, neurons, the microglia themselves, and oligodendrocytes is really important for us to develop safe and effective blood tests as well as new medicines. One other thing to clear up is there's somewhat of a misconception, I'd say, about how easy it is to do this type of work. In the ideal case, this is just a walk in the park, you would say, where you've got kind of 3 steps involved in biomarker development: the sample fluid collection, both in preclinical and clinical models, then biomarker identification, where we use large-scale omics tools, including proteomics, based on mass spectrometry or ImmunoPCR or other methods to identify the biomarkers that you want to measure for different health conditions or diseases. And then step 3, validation of the biomarker and the clinical assays to make sure that you can measure those biomarkers in a large number of patients and prove that they're providing value in terms of aiding decision-making by physicians and their patients. So the reality is that this is a really complicated development path in the real world that is more like hiking up a big mountain. And I can speak to that with some experience. My co-founder, Kevin Wang, pioneered the first FDA-cleared blood tests for traumatic brain injury. We discovered some markers of traumatic brain injury in 2003 and published a paper on that recently called The Game Changer. And it was a kind of a torturous path, and now we are in 2025. And so part of this talk is us trying to do this smarter and faster with some of the newer tools like ImmunoPCR. So Gryphon's partnering with Thermo Fisher Scientific to try to simplify and accelerate this pathway with an ImmunoPCR workflow. It starts on the far left. We're using the proteomic discovery platform from Olink to screen thousands of proteins in brain tissue and CSF samples. And that's because the levels of these proteins are higher in these 2 sample types. There's also less of a dynamic range. That is, that there aren't very low-level proteins in the presence of very high-level proteins that can mask the low-level ones as much as there is in blood. And from this, we try to identify tens to hundreds of biomarker candidates before we take it to the next step, which is the clinical assay development, where we're using the Invitrogen ProQuantum platform. This is a different type of ImmunoPCR chemistry, as you know, but it lets us leverage our diagnostic monoclonal antibody reagents that we've developed to develop assays for our top-ranked biomarkers from our discovery work. And of course, that involves manufacturing reagents in partnership with Thermo, and then developing those assays for different types of biofluid samples, including venous plasma and capillary blood in our case. Then after you've got the assay developed for some of your top biomarkers, you want to do a clinical verification step in a large patient cohort. We've begun to do that, where we're testing, let's say, tens to hundreds of candidates in blood and capillary blood samples from historical patient cohorts. So these are archived samples. And again, we will select the top-ranked biomarkers, including as well as the top-ranked assays before we go to the fourth step, which would be to actually validate the clinical product, that is the reagents, the assay, the instrument, and the software for testing in even larger historical cohorts, but also in samples from prospective patient cohorts that we enroll into a new study. So Gryphon's focus is really on what we call temporal brain biomarkers for traumatic brain injury, but also for Alzheimer's disease. To define this, we were kind of surprised years ago that biomarkers don't just linearly increase as you get older, for example. They have distinct waves of elevated levels and a distinct sequence of which biomarkers are observed. And that's true both in the short term, for example, in traumatic brain injury in the first days to weeks or months, as well as in the long term, the much later stages of traumatic brain injury, decades out from the injury, for example, but also the beginnings of Alzheimer's disease, which can start, very early, depending on the patient population, from 20s to 40s, but generally in your 60s. And if you look at these biomarkers are shown on the right, these are some of the well-known examples like interleukin-6, a pro-inflammatory cytokine, glial fibrillary acidic protein, or GFAP, neurofilament light, or NFL, and phospho-tau. And what you can see is there's definitely a distinct pattern where there's a sequence of these markers. The circles show potential collection points. And so there's something called the Nyquist theorem, which states that you can only measure something if you can measure it at twice the frequency it changes or better. And so if we were able to measure these biomarkers continuously in the patient population, it'd be very easy to define their trajectories or waves. But since in the real world, we have to pick certain time points to collect samples, what you'll see is that you're going to get less of a clear picture on what the trajectory is of these biomarkers in different patients, because you can only sample so many times realistically. And that's both for practical reasons as well as cost reasons. So it's important to really understand the trajectory in the different populations. We focused on TBI and AD. And another way to say this is that you really need to collect the biofluids at the right time and the frequency to catch the wave of the temporal biomarkers in each patient. And this lets you make decisions, for example, in TBI, on whether or not the patient should, for example, be discharged from the ICU or be readmitted or return to work, play, or duty decisions. Fortunate for us, there is strong overlap between the biofluid biomarkers for traumatic brain injury, mild cognitive impairment, or MCI, and Alzheimer's disease. As I said, the steep waves I just showed you in TBI are a bit different in Alzheimer's. It's more of a slow rise as you get older, followed by a precipitous drop in some biomarkers, for example, amyloid beta levels. And sometimes that reflects aggregation of those proteins in the brain. And sometimes it's not clear what it reflects. So the cartoon in the middle is just showing that in an Alzheimer's brain, you have some brain atrophy. In an injured or TBI brain, you might have a brain lesion. But in both cases, you have some common classes of biomarkers like GFAP on the top right from astrocytes, interleukin-6 from microglia and other cells on the bottom left, and of course, p-tau from neurofibrillary tangles, amyloid beta from amyloid plaques, neurofilament light from axonal injury of neurons. And then we're also very interested in mitochondrial proteins like cyclophilin D is in the bottom right. So how can measuring temporal brain markers improve the standard of care for TBI patients or for AD patients? This is just some of the examples. We think this will aid in care and treatment decisions if you can monitor the biomarkers in these patients. It's also known as a personalized medicine or precision medicine approach. And for us, we're particularly interested in the moderate severe TBI patients in the ICU in the first few days after injury. Physicians need to make decisions about intracranial pressure, whether or not to weigh in on life quality and end-of-life decisions. Also, disposition is a major decision. Is the patient ready to be discharged from the ICU, or as I said earlier, readmitted to the hospital? And can they go back to work, go back to play, for example, if they're an athlete, or go back to duty for the military? And all this is based on the disease trajectory that you can get from the biomarkers or the waves. Patient stratification by injury type is a big one as well, particularly for selecting drugs for clinical trials. And often this is based on injured brain cell types. So having a panel of markers that represents different brain cell types is useful for patient stratification. Some of the other things that we'll think about is, of course, early disease detection and diagnosis. Prognostics, are the biomarkers indicating that the patient has a likelihood of good outcome or poor outcome, for example, 1, 3, 6 months, or 10 years down the line. And then for drug development, of course, in addition to clinical studies, biomarkers are very useful for preclinical studies or drug studies in animals and cell models. In the clinic, using biomarkers for patient selection and enrichment of clinical trials, also as pharmacodynamic markers or readouts for the impact of drugs on accumulation of brain cell debris is important. And lastly, having great biomarkers and a temporal pattern for them that you monitor is super useful for AI and generative AI-powered clinical trial design. So this is an example of how we did some discovery work with the Olink platform from Thermo. As Aleksandra mentioned, this is an ImmunoPCR-based method that uses proximity extension assays, which are great for multiplexing. And we actually measured more than 3,000 proteins in cerebral spinal fluid from TBI, MCI, and AD patients. And one thing that we like about this technology is, in addition to be able to measure a lot of biomarkers in small sample volumes, is, as I said earlier, we can really go deep into the lowest level brain-specific proteins that are in the sample of interest. And sometimes that can be challenging by mass spectrometry approaches, but they also have great utility for other reasons, like finding post-translational modifications. So we use both types of tools. And what we found, as you can see, is there's lots of trajectories. Any one of the quadrants shown in this figure is the level of a biomarker in TBI, MCI, and AD patients going from bottom left to top right. You can see a wave for each biomarker, and every kind of wave you can imagine can be observed from these 3,000 proteins. We use this information to select or down select our favorite candidates, knowing that we're looking for biomarkers that go from bottom left to top right, meaning that they clearly increase in levels with cognitive impairment. And that means we've essentially finished our proteomic discovery step that I showed you earlier. So we've screened thousands of proteins in CSF samples, also brain tissue samples, to identify hundreds of candidates. Here I'm just showing examples of 3 proteins that were found in that screen. The relative abundance is plotted on the y-axis, and the clinical dementia rating scale score, or CDR score, of the patients is plotted on the x-axis. And that includes TBI, MCI, and AD patients from left to right, where the CDR goes from 0 to 2, where the red dots represent CDR of 2 for just a few AD patients. And what you can see is that an astrocytic protein that we're particularly interested in had a fairly linear increase from TBI, MCI to AD, although the AD with a CDR2 looks a bit low. But this gives us a lot of confidence that we can use this protein to predict patients that are going to have long-term disability and a risk for death from MCI and AD originating in traumatic brain injury. The pro-inflammatory cytokine is a bit more complicated pattern, as is the mitochondrial protein. This is just an example of down selection to 3 proteins of interest. Once we've down selected we of course then went to the next step in the process, and that's to develop the clinical assay with the in vitro gen ProQuantum platform. And as Aleksandra said, this is a PLA-based technique where you have antibodies binding to two different epitopes on your protein analyte. The antibodies have been tagged with 3' and 5' oligos, where they're ligated together with a splint oligo, and then the signal is amplified through PCR, where you end up with a plot like shown in the top right, where the number of cycles for amplification is shown on the y-axis and the concentration on the x-axis. And so far, we've gotten terrific analytical figures of merit, good linear dynamic range, low limits of quantification, below 10 picograms per mil. And we're also able to distinguish controls from severe TBI patients. That's shown in the bottom right part of the slide for neurofilament light, as well as for the astrocytic protein I showed you earlier. And the p-values show that these are statistically significant differences. So that means that we've selected some good proteins from our discovery work to develop on the ProQuantum platform as clinical assays. And then, in addition to continuing to do that work, we of course then began to manufacture reagents for this with Thermo, and then further develop the assays so that they're more robust. This is just showing some benchmarking data on the ProQuantum platform, neurofilament light on the ProQuantum versus the Quanterix. The R-squared value is 0.985. And as you can see on the correlation plot on the left, very good correlation for the most part between results. We don't always see this with some of the other platforms that we've looked at, so that's great to see. And then again, on the plots on the right, the dots essentially overlap for the two platforms. We've since gone on to begin clinical verification in larger patient cohorts to do some longitudinal monitoring of the temporal biomarkers in different biofluid samples. And for us, we're focused both on venous plasma as well as capillary blood, where the capillary blood is collected on a dried plasma spot or dried blood spot device following a finger prick to collect 3 to 4 drops of blood. And what you can see in this figure is that we are able to, quote, catch the waves of the temporal biomarkers for each patient. And for the capillary blood, this meant that we're likely to be able to do this with out-of-hospital settings. Although it's easy to collect, venous blood or venous plasma in a hospital setting, it's trickier when you've been discharged and you're at home. So we really like this capillary blood collection technology for that application. So what I'm showing in the cartoon here, the video, is that the top 4 panels actually represent the levels of GFAP going down with time for patients A through D. And you can see that although there are some differences in the relative levels of GFAP between, say, the top left quadrant for patient A and the second quadrant for patient B, the trajectories are basically follow the same path. That is, GFAP levels are going down in the first few days following traumatic brain injury. The second row in this figure is the levels of neurofilament light. And again, you can see that for all the patients, the neurofilament light levels are following a specific trajectory. In this case, it's going up with time. And in both the top row and the middle row, you can see that there's some variance, and that's a bit greater in the capillary blood, particularly for patient C in the middle row for neurofilament light. And if you look past the variance, although things are trending a bit, you really don't want that much heterogeneity or variability in your measurement. So we looked at different ways to account for that. And one of the ways we found that was useful was to take a ratio of the 2 biomarkers together, which is a common practice, for example, for amyloid beta in AD patients. So this is the bottom row is showing the ratio between GFAP and NFL. And as you can see, that the curves get a little bit smoother. And the reason for that is if you have, for example, a poor capillary blood sampling event, for example, in patient C at a particular time point, well, it'll be poor for all the markers in that sample. And so there might be lower recovery of GFAP or lower recovery of NFL, but there's definitely lower recovery of both. And it's a systematic error that you can account for by just ratioing them together. So we're still investigating whether or not some of these differences between plasma and capillary blood are real biological differences or simply artifacts of sampling differences. But all in all, we're very excited about this, that the capillary blood and the venous plasma levels look like they're well correlated. I'm looking at some correlation plots now. The top row is, again, the patient's A through D for GFAP with correlations that are strong, and then Neurofilament Light for the second row. And again, for the most part, if we take the ratio of GFAP to NFL that's shown on the bottom row, we improve that correlation. I think that's on to the next one. So we're back at where we started here. This is our platform, our ImmunoPCR workflow. We've screened thousands of proteins with the Olink platform in CSF samples, identified some candidate biomarkers, started assay development on the ProQuantum platform with our diagnostic antibody reagents, manufactured those reagents, developed the immunoassays for them, still doing some work there. But then the next step is really to verify in large patient cohorts, both of traumatic brain injury and Alzheimer's patients. And then ultimately to validate our clinical product with a clinical immunoassay for our top-ranked blood and capillary blood samples. And those would actually be not just from historical cohorts of patients, but also from prospectively collected new samples from new clinical studies. These are just some thoughts of mine about future directions, how this could transform brain health monitoring. I really like the marriage of these two technologies for ImmunoPCR-based proteomic discovery and assay development. And so we'll continue to do this work for a variety of biomarkers and indications, but focused on TBI and AD. I think that some of the emerging technologies like capillary blood sampling have a lot of potential to be highly scalable and cost-effective for brain health monitoring, particularly for out-of-hospital settings. And one way to think about that is that if you decouple the sampling of the biofluid from the analysis, then you're no longer required to make a small point-of-care device for measurement of the biomarker. You can actually send the sample to a centralized hospital laboratory or other lab, and the best instruments can be used to measure the lowest level biomarkers. And I think that's a good, cost-effective and scalable way to do this kind of work. And then lastly, and most importantly, is we want to do all this to improve patient outcomes. And so we strongly believe that if you monitor brain health with these biomarkers, that can aid quantitative decisions by physicians for early detection and better patient management. Also, provide greater understanding of how these biomarkers act as a surrogate for cognitive impairment and the clearance of brain cell debris. Helps make decisions for timely and tailored interventions, including drugs for both traumatic brain injury and Alzheimer's disease. And lastly, since these are protein biomarkers, they can also serve as potential therapeutic targets, which is really exciting and probably the subject of another talk. So thank you so much for your time. Just like to state at the end that this work right now is for research use only and not for use in diagnostic procedures. Appreciate any questions. Thank you.
All right, wonderful. Thank you both again for your very informative presentations. We will now go ahead and move into the live Q&A portion of the webinar. So if you happen to have a question that you would like to ask, please go ahead and do so now. Just type your question into the Ask a Question box and click Submit. We will answer as many questions as we have time for. Okay, let's go ahead and get started here. The first question that we have says, hi, thank you for this presentation. Considering the complexity of neuroinflammatory processes in TBI and Alzheimer's, What strategies do you recommend for selecting the most reliable proteomic biomarkers to advance into clinical ImmunoPCR assays?
I guess I'll take that one. Can you hear me okay? Yeah.
Yes, we can.
Okay. Yeah, I mean, it's a good question. There's so many biomarkers that you could potentially measure and so many in the literature. Where do you start? And in particular, the question asks about what are the most reliable. So what we typically do is try to find what are the gold standard biomarkers in that indication. So neurofilament light, for example, very important across indications. So you definitely want to have some CNS specific markers like that. And then, so for example, interleukin-6 is a well-known pro-inflammatory cytokine. And then once you've got at least the ability to measure those and you can measure them reproducibly, then we venture deeper and look for other more exploratory markers. But I think it's important to take steps so that you're sure that you've got good sample collection methods in place and that you're making those measurements reproducibly.
Fantastic. Thank you. The next question that we have here says, how do you see mass spectrometry complementing ImmunoPCR?
Yeah, I think it's a really interesting question for me. I spent most of my career doing mass spectrometry and love the fact that you can find post-translational modifications of proteins by LC-MS, and it's sequence specific. So the way that we use it is we'll commonly do proteomics discovery work, both with ImmunoPCR and mass spectrometry for brain tissue and CSF, for example. But then when we move into blood, that's plasma or serum or capillary blood, then we will essentially just move forward with ImmunoPCR methods that are better at digging deeper into a wide dynamic range of proteins, where you could have some masking by albumin or the high abundance proteins of the lower abundance proteins. And sometimes that means that we need to develop our own antibody reagents to do these ImmunoPCR measurements of some post-translationally modified proteins that we found by mass spectrometry. So there's a nice back and forth between the two technologies.
All right, wonderful. It looks like the next two that we have here are for Aleksandra. The first question says, do you offer custom assay development for your immunoassays? I have proprietary targets and it might not be in your catalog.
Yes, we as a Thermo Fisher, we do offer custom development services. So if a target is missing, there is you can find the information on our webpage. We will support you during the development process. And as I said, all the information, there is a contact address, can be found on our webpage. In addition to that, there are also additional services that we offer, such as bulk packaging and also lot reservation.
Perfect. And then this next one, Aleksandra says, why is it needed to have different assays for neurofilament light detection and quantification?
As you saw at the beginning of my presentation, we have developed the NFL assays for ELISA, ProQuantum, which are single-plex technologies, but also for ProCaraplex, which is a Luminex technology, multiplex based on the Luminex technology. It is important because in all these three platforms, we showed a really high correlation. That means that if with the complexity of the neurodiseases, as Dr. Haskins mentioned, we need to analyze multiple markers. At some point, we will come to routine analysis where the users will analyze maybe a couple of targets are interested, or a single target technology such as ProQuantum or ELISA. So in that case, they can easily basically switch from a multiplexing to a single target technology because of the high correlation with our platforms. And I also want to add that Also, based on the data that were presented, we can our assays, all three platforms are highly sensitive and able to detect levels not only in cerebrospinal fluid, where NFL is expressed in higher concentration, but also in serum plasma, where the presence of NFL is low.
Perfect. Thank you so much. It looks like the next couple that we have here are for Dr. Haskins. The first one here says, what are useful detection limits for the ImmunoPCR methods used in your TBI and AD work? And would there be a benefit in pushing to lower limits?
Yeah, it's a great question. We generally try to develop assays that have a lower limit of quantitation, somewhere in the single digit range, picogram per mil, so 1 to 10 picogram per mil. Is there a need to go lower to like femtogram per mil? I think the answer is yes. So further away from traumatic brain injury, say decades after injury, it of course gets harder to make measurements. And that's more true for biomarkers that start out at low levels, for example, p-tau, than for markers that are at higher levels, for example, GFAP. So for traumatic brain injury, that's certainly true. We'd like to be able to measure at femtogram per mil levels, but we'd still try to choose more abundant biomarkers to make it easier for us. And then from Alzheimer's, similar answer, we want to measure the proteins at the earliest stage, well before someone gets Alzheimer's disease, 1 to 2 decades before AD onset, for example. And then again, being at femtogram per mil levels works great. So I guess the short answer is I like to put everything in our favor to have the best odds. We tend to choose highly abundant markers and then pair them with the most sensitive detection strategies. But for most of our work, in that 1 to 10 picogram per mil range is usually sufficient.
All right. This next one, Dr. Haskins says, can't lysosomal enzyme be used to disintegrate the brain cell debris?
Yeah, I think this question has to do with the microglial phagocytosis. So the immune cells of your brain, the microglia, are scavenging for brain cell debris like amyloid beta and p-tau, and will engulf or eat that debris and then digest it in the lysosome. But it's not completely digested, and many microglia and other brain cells are injured and then just spill their contents into the extracellular fluid, particularly for brain injury. So you're not necessarily able to just measure the digested components, which could be down, to the amino acid by that point. You've also got intact proteins from cells that died and spilled their entire contents into the extracellular fluid, and then that's cleared into the blood by different mechanisms.
All right. Aleksandra, what is your LLOD for ProQuantum?
So when you look at the different singleplex assays that we have in our portfolio, ELISA and ProQuantum is much more sensitive. That means with a wider dynamic range of the PCR, we can achieve sensitivity in a femtogram range compared to ELISA, where we have a picogram range. So it's much more sensitive assay.
Okay, perfect. And then it looks like this next one is also for you, Aleksandra. This one says your ProCartiplex product. You mentioned it can measure 80 targets, but what is the assay time?
It's around 4 hours assay time.
Okay, perfect. Let's go ahead and move into, we have a few more here. This next one says, are there baseline levels of your selected biomarkers in healthy controls? And are there any similar to troponin where baseline is very low or undetectable?
Yeah, I can take that one. It's a good question because it kind of ties back to some of the other things we talked about. So generally, we find that post-translationally modified variants of some of the more abundant proteins tend to have very high signal to noise, and they are less likely to
be in healthy controls than just the unmodified protein. So we like looking for these variants to get the best signal to noise and the best sensitivity and specificity in our studies, and ideally not be able to measure them at all in healthy controls, because we just want to measure the pathology of brain injury or Alzheimer's disease.
All right, wonderful. So we will go ahead and wrap up with this last question here. This one says, what are some of the criteria you use to select protein biomarkers?
Yeah, I mean, I think I alluded to it in my talk. One of the things we use is the trajectory analysis, kind of what is the time course of the protein biomarker in the indication, or as it crosses over from one indication to another, for example, TBI crossing into MCI or AD. Another criteria we use is we like to have a balanced panel of biomarkers that represents different brain cell types, like neurons, astrocytes, and microglia. And then, of course, there's lots of statistical and biophysical metrics we use to select good biomarkers. Is the assay capable of getting an LOQ in the single-digit, picogram per mil level, for example? But also when we're calculating the area under the curve for predicting outcome, do the statistics show us that really is a predictive biomarker? So I don't have something I can share, but I'd say there's at least 20 criteria that we use when selecting good biomarkers. And, IP is certainly one of those criteria as well.
All right, wonderful. Well, thank you again, Dr. Haskins and Aleksandra, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the information you provided at the time of registration. This webinar can be viewed on demand. LabRoots will alert you when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, everybody, goodbye.
Thank you.